Hello, Dr. Daniel in class. This is my week 6 final presentation on the neurobiology of an axodendritic synapse. The purpose of this PowerPoint is to present a cogent overview of the microprocesses generated within a synapse and to advance the discussion of neuroscience in general. We will briefly discuss cellular anatomy to provide a cursory review in order to add greater context for neural functions and biogenesis, the structure of a neuron, the role of a synapse, and the microelectrical impulses that perpetuate cerebral activity and allow consciousness to exist. Cellular anatomy. It is important to begin with a rudimentary review of the anatomy of a cell, specifically the cell body or soma, so that the discussed microprocesses of a neuron can be extrapolated for a higher resolution of understanding and contextual background. A cell contains many substructures and organelles that generate proteins and enzymes, and, most relevant for this discussion, neurotransmitters. A phospholipid bilayer membrane surrounds the cell and bifurcates the intracellular environment where most biogenic synthesis is generated, and the extracellular space, which is the environment located outside the cell's membrane. One of the principal organelles located inside of the intracellular space is called the mitochondria. This structure generates adenosine triphosphate, or ATP, a neuron's principal energy source to facilitate its functions. Moreover, the Golgi apparatus is an intracellular organelle that encloses neurochemicals produced by the cell into small, consolidated compartments used to transport endogenous materials through the cell. This will become more significant later in the presentation, especially concerning the exocytosis phase of a presynaptic neuron. Next is the structure of neurons, the nerve cells found in the central and peripheral nervous systems. The soma, the body of the neuron, is where the aforementioned cellular endogenous processes that produce biogenic materials take place. Proteins and neurotransmitters are synthesized here, and they are the primary chemicals that facilitate neural activity. The dendrites are tentacle-like structures attached to the neuron's soma and are used to receive chemical signals from another adjacent neuron. The axon of a neuron is the limb connecting the soma to the axon terminals and is the channel by which the electrical signals called action potentials are propagated. The axon terminals are the structures located at the end of the axon and are responsible for sending signals, neurochemicals, to another neuron for transmission. The neuron essentially communicates and transmits signals to the nervous system that actuates various biofunctions such as breathing, speaking, thinking, and processing information. Moving on, we will discuss the function of a synapse. The synapse describes the contact area between two neurons that send and receive neurochemicals to actuate various signals for neural processes. The presynaptic terminal, typically the axon terminal, which was formerly discussed, releases the neurochemicals. Depending on the availability of receptors and ionic stimulation of the extracellular and intracellular environment, these chemicals will pass to the postsynaptic terminal, the dendrite, through a sundry of ion-gated channel receptors. The leftover released neurotransmitters will either reabsorb back into the presynaptic terminal, diffuse out into the extracellular environment, or break down into their subconstituent components through a process called enzymatic degradation. This is essentially the waste management and recycling processes of the biogenic materials of a cell. Let us look deeper at this diagram, which is the summit of this presentation, and work through the synaptic processes of the axodendritic synapse. This image of an axodendritic synapse demarcates the connection of a presynaptic axon terminal and the postsynaptic dendrite. First, an action potential, which will be discussed in the upcoming slides, is generated and travels to the end of the presynaptic neuron's axon terminal. This electrical impulse signals the release of neurotransmitters through a process called exocytosis, facilitated by high concentrations of positively charged calcium ion molecules within the intracellular environment. The synaptic vesicles, which contain the neurotransmitters initially packaged by the Golgi apparatus, fuse to the intracellular presynaptic membrane and are released into the synaptic cleft. This gap, or cleft as it is called, is the small extracellular space located between two neurons. From the gap, the biogenic materials will pass through their corresponding ion-gated ligand channel receptors located on the outer membrane of the postsynaptic terminals. For example, glutamate will pass through specific glutamate receptors designed to receive glutamate molecules. Likewise, gamma-aminobutyric acid, or GABA, will pass through its specifically designed ion receptor for chemical transmission. Lastly, surplus neurotransmitters accumulating in the synaptic cleft will reabsorb to the presynaptic reuptake receptors, or molecular deconstruction will occur through the process of enzymatic degradation, as mentioned in the previous slide. Action potentials. An action potential is essentially an electrical current. This current acts as the impetus of neural signaling momentum and propagation through the axons, generating system inductions from one neuron to the next. Action potentials are a critical part of a synapse, as they govern the type of messages transmitted and received by neurons. These action potentials are initiated by ionic diffusion, characterized by molecules that occupy an environment of high concentration moving to an environment of low concentration. For example, and with the help of electrostatic pressure combined with diffusion, when the intracellular domain is populated by high concentrations of positively charged potassium ions, negatively charged chloride ions diffuse out from the intracellular space into the extracellular environment through their appropriate ion-gated receptors. This results in a depolarization of the intracellular environment characterized by higher concentrations of positively charged ions. The occurrence of depolarization of the intracellular space is the trigger by which an action potential is generated. Excitatory and inhibitory effects. These action potentials can carry either excitatory or inhibitory neural responses. An excitatory response can increase the probability of generating an action potential. This will ultimately determine if the action potential will propagate from neuron to neuron by effectively sending and receiving neural signals through the nervous system. Inversely, an inhibitory response will decrease the probability that an action potential will occur, thus nullifying any propagation of signals by the neuron. It should be noted that specific neurotransmitters produce such messages. For instance, glutamate is a neurochemical that possesses excitatory stimuli, and neurons that receive graded concentrations of glutamate ions will receive excitatory messages, thereby generating electrical impulses. Moreover, GABA is an inhibitory neurotransmitter that abolishes the production of action potentials. The neurochemical influences of synaptic action potentials are important to subsume because they ultimately determine if neural activity is actuated. Conclusion. In summation, the neurobiology of an axodendritic synapse is vast and complex, with molecular and cellular transactions beyond measure. This presentation attempts to elucidate the multidimensional machinations of cells, neurons, and synapses to glean a deeper appreciation of cellular neuroscience. To recapitulate, neurons produce neurochemicals through the internal biosynthesis of cellular organelles such as the nucleus and mitochondria. These neurotransmitters potentiate either excitatory or inhibitory signals used to transmit neural information. The neural messages actuate sensory and biogenic purposes for cerebral activities, autonomic, cognitive, and interpretive functions. Thank you.